So good day to everyone. My name's Tomasz Jagielski, and it gives me a great pleasure to welcome you all to the FEMS Microbiology Reviews Webinar on exploring the molecular aspects of microbial biology. A word about myself, if you allow. I work at the University of Warsaw. I head the Department of Medical Microbiology. I had this very special privilege to serve as the editor of the thematic issue of FEMS Microbiology Reviews, entitled Exploring the Molecular Aspects of Microbial Biology. This issue is conceived as the closing event of the 2022 celebrations marking the 100th anniversary of the birth of Louis Pasteur, the father of microbiology. The issue also served as a kind of postscript to the conference that I organized in late November 2022 in Warsaw, dedicated to the Pasteur's Jubilee. A few snapshots from this event you can see now in the welcoming. Many contributors to this special issue were distinguished guests. The meeting and delivered their talks. So once again, I cordially welcome you to this webinar, which can be regarded as a symbolic, let's say, final quote in this concert in honor of Louis Pasteur. I had the honor to co-chair this today's webinar with Professor Grzegorz Wegrzyn from the University of Gdansk, who is a regular editor for the FEMS Microbiology Reviews journal, and is also an eminent microbiologist and one of the most internationally recognized scientists from Poland. So Grzegorz, it's great to have you here. Let me now convey a few words on the mission of FEMS, the Federation of European Microbiological Societies. So briefly, this mission's purpose is to champion science and deliver impact. FEMS fosters scientific communities, providing different opportunities for microbiologists to connect in person, online, and through the journals. So we, as part of this society, support the next generation of microbiologists through meetings, webinars like this one, summer schools, grants, and many more. FEMS advances the microbiology community through magnifying microbial impact. We use the income from our journals, and this is quite important, to fund different activities and support the community. Yeah, and our today's webinar will showcase leading authors of reviews that span Pseudomonas taxonomy and evolution, fungal phenotypic diversity, or molecular epidemiology and evolution of Mycobacterium, tuberculosis. These authors, today's speakers, are top experts in their fields, and I will give a few more words when introducing each of them individually as we go. And I want to direct our audience to please present their questions to our authors by using Q&A function at the bottom of our screen. And we will select some questions to ask the authors during the discussion panel at the end of all presentations. So I think we will start. Our first lecture will be shared actually by two speakers. First will be Dr. Zulema Udaondo, who works as a research scientist in the department. Yeah, is that correct name, or are you using perhaps someone's
No, it's correct.
Yes. Oh, thank you.
Thank
I
you.
know. Sorry. It's perfect. So, hello, Zlema.
Hello.
She works as a research scientist in the Department of Biotechnology of the National Center for Biotechnology in Madrid. She trained at Spanish National Research Council and the University of Arkansas. She's an expert in bioinformatics and leads the pan-genome research area. Her broad approach can be used for unequivocal species identification in microbiology, as well as for the identifying core genetic functions that drive physiological and niche colonization. And immediately after Dr. Udaondo, the lecture will be continued by Professor Juan Luis Ramos from Estión Experimental del Saidín of the Spanish National Research Council in Granada, if I pronounce it well. So let me just tell a few words about Professor Ramos. Without exaggeration, I can say that Professor Ramos is among the most distinguished microbiologists working today. He has deeply expanded our knowledge on microbial physiology, genetics, and molecular ecology, and applied it in order to come up with better ways of restoring polluted sites using microbial biodegradation. And Professor Ramos, it has to be stressed, is laureate of the Lvov Medal of FEMS in recognition for his contribution. He's also a member of the American Academy of Microbiology, the European Academy of Microbiology, and the Granada Academy of Sciences. And both Dr. Udaondo and Professor Ramos will talk about the genome diversity of the Pseudomonas putida group, covering its taxonomy, pan-genome and core genome structure, and antibiotic resistance mechanisms. With that said, I invite Dr. Udaondo to begin. Oh, yes, we see your presentation. So, yes.
Thank you. Thank you so much, Tomasz, for the introduction, and hello, everyone. Thank you so much for being here, and to the organizers for inviting us to share our work with all of you. And today we will present part of our paper that was published in December 2024, in which we explored the genomic diversity, the taxonomy, and the population structure of Pseudomonas putida group. This was a joint effort with Professor Ramos and Dr. Caleb Abram, who is in the audience, I think. And my role today will be to guide you through what these strains really are at the genomic level and why this matters. And then Professor Ramos will continue with the mechanistic aspects of solvent tolerance, focusing on Pseudomonas strains. So first, what are Pseudomonas putida strains? So Pseudomonas putida are soil microorganisms that are very interesting from both an ecological and a biotechnological point of view. And this is because some of these strains are well known for their ability to promote the growth of plants through a variety of direct and indirect mechanisms that I have tried to summarize here. However, some of these members of this species are remarkable for their capacity to tolerate high concentrations of organic solvents, which are toxic compounds for most living organisms. Because of these different properties, because the ease of manipulation of these strains, and because of the high versatility, the high metabolic versatility of these microorganisms, so the Pseudomonas putida strains have been used in different applications, such as, for example, resource remediation of contaminated soils, which consist of removing pollutants from the rhizosphere, which is the area of soil that is surrounding the roots of the plants and that is under the direct influence of the root exudates. Also for the production of biofertilizers and other added value compound in co-cultures and symbiotic systems, and also in genomic studies for growth improvement. So these are some classical strains that have been historically classified as Pseudomonas putida. We have, for example, Pseudomonas putida Ver1, which is a plant growth promoting rhizobacteria resistant to desiccation, and that has been successfully used as a biofertilizer. Indeed, you can see in this In this picture, the differences in the development of the primary and secondary roots of this corn seed that was grown in a media without and with bird-1. And you can also observe the differences in the growth of these plants that were grown without and with phosphogel, which is the biofertilizer that was formulated using bird-1. And we have also Pseudomonas putida.t1e, which is a solvent-tolerant strain that thrives in the presence of high concentrations of monoaromatic hydrocarbons. And indeed, this strain is considered as an extremophile because it can stand very high concentrations of these toxic compounds. And because of these properties, this strain has been used for resource remediation purposes. It's been studied for this purpose. We also have Pseudomonas putida HB3267, which was isolated in a hospital environment in France, and that is able to kill insects and is resistant to a large number of antibiotics. And this is because this strain has this plasmid that is called PC9 that codifies for a large number of antibiotic resistant determinants. So, as you can see, at least phenotypically, these strains behave very differently and thrive in different habitats and have very different lifestyles. So, this leads to an obvious question: are these really members of the same species? And this is a question that has been also raised by other research groups around the world, as you can see in all these papers that have been recently published proposing the reclassification of these strains. And this is because rather than being a single homogeneous species, Pseudomonas putida has long been suspected to represent a complex of closely related strains commonly referred to as Pseudomonas putida group. But of course, all of these reclassification raise even more questions because if these strains are not members of a single species but instead represent a set of closely related organisms, then who are the real Pseudomonas putida sensu stricto strains? And what traits actually define these Pseudomonas putida sensu stricto strains? Are these the plant growth promoter rhizobacteria like BER1, or are these extremophiles that can stand high quantities of monoaromatic compounds like .T1E strain? So in order to answer these questions, we knew that we couldn't use traditional typing methods such as cysteine ribosomal RNA sequences or multi-locus sequence typing because these methods lacks the resolution needed to discriminate among these closely related strains at the species level. So for this reason, we decided to design our own pipeline that leverage whole genome comparison techniques at the genus level in order to obtain the population structure of Pseudomonas putida group. So in simple terms, what we did was instead of focusing on a few marker genes, we decided to compare the entire genomes of thousands of Pseudomonas strains using whole genome wide distance measurements. And then we apply unsupervised clustering on these measurements to group strains based on their overall genetic similarity. And by doing this at the genus level, what we aim was to define species level clusters, and then we will be able to characterize them functionally using pangenome analysis. So this is the pipeline that we use in order to obtain species level clusters. We started with a very large data set of more than 26,000 genomes that were labeled as Pseudomonas from the NCBI. So we download all the genomes labeled as Pseudomonas from the NCBI. And then we use GRAMS pipeline. That GRAMS is the pipeline that was designed by Caleb Abram at his PhD thesis project. And at that time, Caleb was a PhD student at the University of Arkansas for Medical Sciences. And MASH uses genetic distances and also unsupervised machine learning and other statistics to group genomes by relatedness. Then we obtain this species level clustering using already well-established thresholds that I'm showing you here, like MASH 0.0 lower than or equal to 0.05, that is equivalent to an 95% of ANI. So these allow us to identify a total of 435 species level clusters across the genus, but in order to validate taxonomically all our species level clusters, meaning with this what I mean is that in order to label all of these species level clusters, what we did was to identify all the type strains within each of these species level cluster. We assign them following the principle of priority in those cases in which we found more than one type strain. For those species level clusters that we couldn't find a type strain within them, we labeled them as putative novel genome species that should be further investigated or classified using polyphasic approaches. So once we got all our species level clusters identified, we then look for all the strains in the whole dataset that contain strains labeled as Pseudomonas putida. We wanted to find how many of these species level clusters contain strains labeled as Pseudomonas putida, and we found that these strains were scattered across 31 different species level clusters, meaning that there is a massive misclassification of these strains in public databases. So among these 31 species level clusters that contain genomes labeled as Pseudomonas putida, then we focus on these of 9 cliques or species level clusters, and sometimes we call them cliques because of Graham's methodology, but I'm talking about the species level clusters. So we focus in these 9 species level clusters that included at least 6 of these genomes that were labeled as Pseudomonas putida. And as you can see here, we were able to identify genome species that have been very well characterized, such as, for example, Pseudomonas putida monteili and Pseudomonas putida sensu stricto, but we were also able to identify species level clusters from species that have been recently characterized, such as, for example, Pseudomonas asiatica, wariconensis, Guayamba palatensis, and we could also identify 3 novel genome species. Among these 9 species level clusters, 2 of them were particularly important for us, that were species level cluster 2 or clique 2, which corresponds to the real Pseudomonas putida sensu stricto strains, because it contains the type strain for the species, that is Pseudomonas putida MBRC 14164, along with other 40 closely related strains. So this will be the Pseudomonas putida sensu stricto strains. Also, we were very interested in this clique, the species level cluster 1, that corresponds to Pseudomonas alloputida. And this is because this cluster indeed contains the highest number of strains that have been historically labeled as Pseudomonas putida. In fact, these clusters include many of the strains that have been extensively studied over the years, such as Pseudomonas putida KG2440, Pseudomonas putida ver1.T1E, Idaho, and Pseudomonas putida S12. However, when we look at the genomic relatedness of these strains, these are clearly more closely related to the type strain of Pseudomonas alloputida than to the type strain of Pseudomonas putida. So this led us to a striking conclusion, that is that we actually know more about Pseudomonas alloputida species that was recently described than about Pseudomonas putida sensu stricto. But we decided to move on, and once we defined the species level clusters based on population genomics, the next step was to ask whether this taxonomic classification was also supported at the functional level. And in order to address this, we performed pangenome analysis at the Pseudomonas putida group level. And we did this because we used the core genome, that are all these genes that are present in all the strains of the analysis, that were 393 core genes or proteins in this case that we used, that were present in the 413 Pseudomonas putida groups. So we used this core genome in order to construct this maximum likelihood tree that you can see here. We were pretty happy with it because, as you can see, in this tree, the tree shows a clear separation between the 9 species level clusters. You can see here Pseudomonas alloputida, here Pseudomonas putida clustering with other groups, with other species level clusters with these two novel genome species and with Pseudomonas monteili. And of course, we did other analysis. As you can see here, for example, we leverage a matrix, a pangenome matrix of present absent of gene families, and we build this PICO-A plot in which you can appreciate again the separation of Pseudomonas alloputida species level cluster, and again Pseudomonas putida clustering with the two novel genome species, Pseudomonas monteili, and the other species level clusters. So both genome structure and the gene content independently support the same species level classification. Then, after validating the taxonomy at the group level, we zoom at the species level of these two interesting species level clusters. And we apply again population genomic analysis to these two species level clusters, to Pseudomonas putida and Pseudomonas alloputida. And we combine this analysis with all the available metadata that we could find from these strains because we wanted to explore potential ecological differences between the two species. In summary, what we observed was the presence of different clusters that you can see here of closely related strains that are highlighted with these deep blue colors. And these clusters of strains are strains that are very similar between themselves than to any other strain of the analysis, and we classically refer to them as phylogroups. But the important thing is that we observed that these strains were usually isolated also from the same environment. Indeed, what we observed analyzing the metadata is that in the case of Pseudomonas alloputida, the strains were mainly associated with contaminated soils, wastewater treatment plants, and marine sediments. In contrast, Pseudomonas putida looks like displayed a more homogeneous population structure with most of the strains isolated from the rhizosphere, the bulk soil, and sediments. And this analysis suggests that Pseudomonas alloputida could be occupying a broader range of ecological niches, while Pseudomonas putida appears to be more ecologically constrained. And finally, we focus on Pseudomonas alloputida and species level cluster 1 because we wanted to see if this solvent tolerant trait was general of the species. And we found that solvent tolerance is not a generic species level trait, but rather an adaptive feature of specific lineages within the population. And this is something that you can see here in this PICO-A plot that we constructed using again the present absence of gene families, but in this time at the species level using only the pangenome of Pseudomonas alloputida. And in this PICO-A plot, you can see the strains separating into distinct groups that are associated with different habitat preferences or lifestyles. For example, all of these strains in brown colors, they belong to human samples. Here in purple, you have all the strains that are similar to .T1A that were isolated from wastewater treatment plants or from contaminated environments. And here in light blue, you have all these strains that were similar to B1, and therefore they are rhizospheric microorganisms. And here in pink and little bit isolated, you have all your KT2440 related strains. So, with this population level framework in mind, Professor Ramos will now explain the molecular mechanisms underlying solvent tolerance in these strains.
Thank you, Zulema, for the overview of the Pseudomonas putida complex. As a general rule, we can say that the species pangenome consists of core genes that define the basic physiology of a species, accessory genes that are related mainly to niche adaptation, and unique genes that confer specific traits to the strains. Pseudomonas T1 is characterized by its ability to grow in the presence of extremely toxic compounds like styrene, xylene, ethylbenzene, toluene, etc. As you can see here, Pseudomonas T1 can reach turbidity higher than 2 units when growing in the presence of 1% of toluene. This is so remarkable because a single drop of toluene, for instance, in a culture of Escherichia coli, will kill immediately all the cells. So the question is, why is this strain so solvent tolerant? And to approach this, we took a series of complementary approaches. In one of them, we use mini TM5 mutagenesis so that we generate a collection of near 10,000 independent TM5 insertion in the genome. We sequenced this insertion, and we took all the strands into a robot to look for solving sensitive clones and so on, identify genes related to solvent tolerance. In addition, in the next, in this overhead, we took parallel studies doing transcriptomic and proteomics. And what we basically did was to compare the level of gene expression in RNA level between cells growing in the absence and in the presence of toluene. And the same was done for proteomics. So we grew the cells in the absence and in the presence of solvents and identified the proteins that were differentially present in the cells. To make a long history short, I can say that the DOT1 is resistant to solvents because it combines three different mechanisms. One is impermeabilization of the cell membrane, mainly through a very fast cis-to-trans isomerization of fatty acids in the membrane. A second, this impermeabilization is not perfect, so some of the solvent will enter the cell, so the cells induce chaperone to refold and fold the proteins, and also stimulate generation of energy, and at the same time avoid oxidative stress. And finally, what is extremely relevant and unique in these trends is the use of several efflux pumps to remove solvents from the cytoplasm, the inner membrane, to the outer medium, and from that use an inner membrane protein, a channel, and a porine. So these pumps are critical for solvent tolerance. To identify the specific role of these pumps, what we did was to generate mutants in the TTG-ABC operon that is encoding one of the pump, and the GHI operon that is encoding another relevant pump, and we tested solvent tolerance. What we found is that these pumps are able to pump out many different compounds like toluene, styrene, silenes. This is also true for the TTG-ABC. It's able to pump out all these aromatic compounds, but it's also able to pump out flavonoids and mainly antibiotics like ampicillin, chloramphenicol, and tetracycline. These pumps are induced in response to these toxic compounds. And what we found is that the adjacent but divergently transcribed are the regulators of these efflux pumps. All these regulators, TTGR, TTGT, TTGV, all of them are repressors of the pump. So they are somehow, some way inhibiting the transcription of the gene, and when are in the presence of the inducer, the pumps are expressed. To concentrate on one of the regulators, but we have done with all of them, what we did was to crystallize the regulators. This is a TTGR, that is a dimer, is a symmetric dimer. It has a DNA binding domain made of this alpha-2, alpha-3 helix-turn helix that is connected through alpha fold with the central cavity made by the intersection of the two dimers. This TTGR is a repressor, and it binds to the operator region of the TTG-ABC operon, recognizing an inverted repeat, and the protein sit on this motif and repress the expression of the efflux pump. To demonstrate that TTGR is binding to DNA and that different compounds will interact with the regulator and release from the target DNA, we run these EMSA assays. Here in lane 1, what we have is the promoter segment labeled with P32, and this is the free form of the DNA. When to this DNA we add the purified TTGR protein, what we have is that the TTGR bind to the DNA, and there is a shift in the chromatogram. And then from line 3 to 13, what we have is the TTGR present plus a low concentration of different compounds. As you can see, different compounds with different structure, like for instance, naphthol, butylparaben, fluoretine, naringenin, quercetin, that are flavonoids. All of them are able to bind to the TTGR protein. The protein is released from the DNA fragment, and you can see the free band returning to its position. To further analyze this complex response, what we did was to co-crystallize the TTGR protein with different effector molecules. Here I show two antibiotics, tetracycline in the B panel, chloramphenicol in the A panel, and different flavonoids in the other panels. I cannot go into the details, but from the structure of the effector molecules and the surrounding, what we can see that the ability to recognize different effectory, because different residues in the active pocket of TTGR can interact with the active lateral side chain of these effector molecules. And the results of this interaction is always the same. There is a signal sent from the cavity through the alpha-4 domain to the helix-turn helix DNA, so that the helix open and the TTGR protein is released and the efflux pump are transcribed. So solvent tolerance and antibiotic tolerance in DOCT1 are linked through unique efflux pump and regulators that enable a coordinated response to these toxic compounds. Thank you very much, and to all the audience, and also to the funding agency that support our work. Thank you.
Thank you very much, Professor Ramos and Dr. Udando for your enlightening presentations. I can see some questions that need some expert attention, and I encourage you and also other speakers to perhaps respond to them in the chat windows to directly to individuals who submitted them. Okay, and in the meantime, let's move on to next lecture. And the next speaker, who is Łukasz Kozubowski. May I ask for changing the slide, perhaps. Can you load your presentation? Okay, let me introduce Łukasz. He's an associate professor in the Department of Genetics and Biochemistry and the Eukaryotic Pathogens, Innovation Center at Clemson University in South Carolina, USA. His group focuses on defining the mechanism that allow Cryptococcus neoformans to proliferate in the host and to acquire resistance to antifungal drugs. I believe that we all know that Cryptococcus neoformans is an important human fungal pathogen listed quite recently by WHO as a critical top-ranking priority pathogen. So this matter is of utmost global concern. Please, Łukasz, the stage is yours, if I may say.
Thank you very much. So first, I would like to really thank the FEMS and the organizers for giving me this opportunity to speak today and present this work, actually, which is review essentially on a subject matter that is incredibly complex. So in this presentation, I will really try to keep it simple and highlight only some aspects that I personally believe are important. But I also want to point out that I will focus in this case on two organisms that the authors' labs investigate just for illustration. And of course, invariably, I will not be able to acknowledge the incredible amount of work that has been done on this subject and many publications and authors. So I would like to refer the viewers to, of course, our review, but also a number of excellent reviews on the subject that includes fungal species, but also non-fungal, because the subject is really relevant to many organisms beyond fungal biology and most prominently bacterial species as well, but beyond that as well, even higher eukaryotes. So I would like to start with a very simple question, which is what is the phenotype of the cell and how many features cell generally contains. And this is relevant to the question of phenotypic heterogeneity, as you will see. So in principle, we can say that living cells have some few major characteristics that are common to all living cells, like cell membrane, DNA, ribosomes, for instance. They have some features that we all recognize, which are common, which are also specific to some cell types. But in reality, when we think about how many features a cell may contain or have, this is really a number that it's impossible to estimate, and this is because of the huge complexity of genetic networks, expression networks, metabolic pathways. So it's a huge number, essentially astronomically huge. So let's focus on one single specific phenotype of a cell that I've represented here in that kind of a silly way. But I want to make a point that when it comes to a specific phenotype, we would all agree that it is dependent for sure on and determined by the genome. We all know that it also depends on environmental factors. But the topic of today's talk of mine is the contribution of phenotypic heterogeneity as something that is apart from genome and environment, additional factor that shapes the phenotype. So that creates a possibility of various phenotypes in an isogenic population of cells that have the same genome and are even exposed to the same environmental conditions. So I would like to present this sentence because this is the quotation from one of the reviewers that our paper received. And I really like that idea. That's why I would like to post it here. So the reviewer said that the definition of phenotypic heterogeneity is so deceptively simple and yet to and yet how phenotypic heterogeneity can manifest itself and the underlying mechanism appear to be almost arbitrarily complex. I cannot agree more with this statement. When we think of phenotypic heterogeneity in the context of multicellular organisms like our cells, for instance, it's kind of intuitive that, yes, of course it exists. Cells have the same genome, but they are vastly different morphologically. So certainly that phenotypic heterogeneity is there, and it's not very surprising. And we can think of it as some kind of evolutionary advancement, like 700 million years ago, and when the organisms evolved as multicellular organisms, presumably, this was some kind of advancement in terms of their adaptation, and so it can be explained by this allowing the organisms to adapt and form specialized tissues that would adapt those organisms to some ecological niches. But yet, despite all those advancements of multicellularity, unicellular microbes prevailed and continue to thrive in the environment, which is interesting. And even more interesting, those in unicellular organisms still exhibit phenotypic heterogeneity. So the question is, from the perspective of those unicellular organisms, what are the mechanisms and what contributes to that phenotypic heterogeneity? And here I just want to relax a little bit of this term, and rather than focusing on phenotypic heterogeneity as such, that is something independent of DNA, look a little bit more broadly for a moment and think of just phenotypic variability that certainly overall is indeed dependent on the DNA and the genome, and even in the context of initial genome content will change with, let's say, change of SNPs or like single nucleotide polymorphism or some aneuploidies and transposition, for instance, to name a few events. But it also is dependent on at the physiological level, which is non-genetic. And here we can think of some deterministic sources, like, for instance, obviously cell cycle stage for the cell would determine its current or phenotype at the given moment, replicative age of the cell or metabolic cycles, for instance. But there are very interestingly also a lot of sources at the gene expression level, which are mostly considered stochastic sources of phenotypic heterogeneity, like, for instance, the gene expression noise, which can be differentiated as intrinsic or extrinsic, as well as some other random factors that may happen in the cell, like, for instance, during cell division would be partition of certain cellular factors between the dividing cells. So the question also arises, like in the multicellular organisms, we can kind of intuitively think of what is the sense of this phenotypic heterogeneity, obviously, but what about those unicellular organisms? What is the advantage of phenotypic heterogeneity for such communities of cells? And of course, first thing that comes to mind is this ability to withstand stress or react properly to stress. And that is something that probably is one of the prominent aspects of this phenomenon. So to illustrate this, or to give some kind of context in terms of in unicellular organisms, let's consider this population, very hypothetical population of unicellular cells, which exhibits no phenotypic heterogeneity. Arguably, such thing cannot exist in nature. Obviously, there will be always some heterogeneity with respect to some phenotypes, but let's just think of that. What would happen hypothetically? Hypothetically, such population would be very weak in terms of responding to changing environment, and there will be always some conditions that those cells would just simply not withstand, and the whole population would perish. In contrast, if there is some level of phenotypic heterogeneity in the isogenic population, and if it's even low, at least gives some chance for some cells in that population to when encountered some limited number of environmental conditions that are adverse, perhaps some cells would survive. And of course, the extreme would be a very high level of phenotypic heterogeneity, which gives those cells, that population, very high chance to overall survive in the ever-changing environment. So one phenomenon that is really tightly linked to phenotypic heterogeneity is this concept of bed hedging, and the mechanism that is very widespread and thoroughly described in bacterial cells, and more being described now in also fungal species, which is basically the risk spreading strategy that in the isogenic populations can happen, and it just helps to survive in the fluctuating environments. And more rigorous or more the definitions that really are derived from population genetics point out that it is associated with the decreased arithmetic mean fitness and the increased geometric mean fitness of the population across environments. So one example I want to show you here, because I think it's very striking, is the yeast Saccharomyces cerevisiae from this study by Levi and et al. And in this case, what the authors did, they expressed one of the stress response or stress-related proteins, which is the trehalose synthase regulator. They expressed them in the cells as GFP, and they monitored under the microscope the culture. What I want to draw your attention to is that one single cell that is here pointed by the arrow, that single cell will express slightly higher levels of that stress-related protein. And while initially the culture will be growing better for other cells that have low levels, after heat shock, which was briefly shown here, the culture that expresses actually the higher level of this stress-related protein is the winner, essentially. So that very beautifully illustrates the strategy that the population of cells uses by heterogeneity, essentially, of this phenotype, which is the expression of this particular stress response protein, gives an edge to few cells in the population that can just now prevail and grow, while the others, unfortunately, cannot. So another thing I wanted to point out is that while there is a lot of work that to point to high heterogeneity in terms of different colonies that are formed, and that includes bacterial species as well as very prominently in fungal species, less attention has been devoted to the phenomenon of heterogeneity within the colony, and there are some studies out there too, so it's still a relatively studied subject, but relatively less so than the colony morphology within the population. So here what we see is the study by Treven et al., where they express one of the proteins that prominently as GFP, that prominently, and this is a cross-section of the one single colony, what we see here, and very prominently that protein is expressed at the very cell surface, so in the colony surface, by the cells that are on that surface of the colony. And what that allowed the authors to do is now use FACS analysis, essentially sort those cells from a mixture of colonies. They could sort those cells and transcriptionally profile the cells that are outside of the single colony versus those that are inside. And they found very interesting findings that there's really a huge degree of specialization within the colony, single colony. So this is an example of high phenotypic heterogeneity that is associated with colony growth. And this has high relevance to fungal pathogenesis when we think of, for instance, biofilms during, let's say, Candida albicans infections. So I think it's a very interesting subject and requires further attention. So no doubt that phenotypic heterogeneity plays crucial role in fungal pathogenesis and really impacts antifungal therapies. And I just want to highlight few examples of this. And first, I would like to introduce the Cryptococcus neoformans, the subject or the pathogen that our lab works on. It's an environmental fungus that can unfortunately reside in humans, and but this mostly mostly happens in the immunocompromised individuals. So the spores, when inhaled, they reside in lungs, and unfortunately, they can cross the blood-brain barrier and get into the brain and then cause cryptococcal meningitis, which, if untreated, is fatal. So definitely very important subject to study and improve therapies for cryptococcosis. I want to emphasize one aspect that relates to phenotypic heterogeneity in Cryptococcus, which is the very interesting array of morphotypes that Cryptococcus can acquire. In addition to being just a regular yeast cell, it can form a seed cell, which is the very small cell, as well as the giant cell, that is very large size cell, sometimes like even 100 microns across. And what is the advantage of this? Why those cells but or what is the impact on the infection? Is that, for instance, the big Titan cell is so huge that in this illustration seen here is the pseudo-colored pink cell, as opposed to blue regular size yeast that is engulfed by the macrophages in the lungs. So clearly, this Titan cell is so huge that it just simply cannot be engulfed by the macrophages. So it's a clear defense mechanism, so to speak, when it comes to Cryptococcus. And on the other hand, when it comes to tiny little seed cells, what has been proposed by, for instance, Jessica Brown in her lab, is that the seed cells are so tiny that they can, in the initial infection in the lung, and what's shown here is the alveolus, lung alveolus in that schematic, which contains all sorts of morphotypes of Cryptococcus. Those tiny cells are so tiny that they can actually now easily penetrate through the blood vessels and get disseminated in the other parts of the body. So there's a lot of roles that those morphotypes can play during infection, which seems to be critical for infection and prevents potentially effective therapies, but also gives us a chance to think of ways to treat cryptococcosis. And the last thing I want to point out here is the role of phenotypic heterogeneity when it comes to drug treatments and antifungal drugs specifically. And one prominent example is fluconazole, which is basically the azole drug that is used very often during many fungal infections, including cryptococcosis as well, although it is not a drug of choice for cryptococcosis because it usually doesn't kill. There are other drugs that kill cryptococcus, but it's nonetheless used as the maintenance therapy or in areas where those more effective and unfortunately more expensive drugs are not available. So fluconazole inhibits ergosterol, and by doing so, it inhibits cell growth. What is important here is that while it is effective in inhibiting cell growth, it is not very effective in killing, and that gives the cells chance to potentially survive the treatments with fluconazole. What I'm showing here is the classic disk diffusion assay that very nicely illustrates the heterogeneity within the population and how, in this case, candida cells can react to fluconazole. In this assay, the drug is the cells are spread uniformly on the agar plate, and then the drug is placed in this tiny little disc that is placed right in the middle of the plate. And after a few days, or in this case 24 hours, actually already we see vast, like majority of cells away from the disc just grow. But where there is a gradient of the drug that goes from the disc outside into the culture, we see a zone of inhibition. There's no growth because the cells are basically affected by the drug. And the disc, this kind of zone of inhibition, defines the theoretical MIC, the minimum inhibitory concentration of the drug. Now, what we see is that after 48 hours of such growth in Candida albicans, we see gradual growth or kind of slower growth within the zone of inhibition. So while general population is affected by the drug, there is still some cells in the population that slowly grow into the zone of inhibition and are able to withstand the pressure or the inhibition that the drug imposes. So when we think about the probability that the cells will actually grow in the zone of inhibition or in the presence of the drug. We can imagine few case scenarios here. The most simple one is if essentially all the cells in the population can simply grow in the presence of the drug, they are completely resistant. There is no variation here, and the cells, even to the maximum achievable concentration, just all grow. The probability is just nearly 100%. On the other hand, if the culture is susceptible to certain achievable concentration of the drug, we will see some MIC. We will see the cells. We'll see very clearly defined zone of inhibition when the cells just stop growing, and that will define this minimum inhibitory concentration. The heterogeneity is evident when we consider populations that actually are heterogenic, and they show some kind of level of tolerance. And in those cases, we see two cases where the tolerance is there, but it's low on the left, or tolerance is much higher on the right. So while majority of cells will grow till the MIC concentration, there's still some subset of population of cells which will grow in the presence of the inhibitory concentration of the drug, and these cells will be tolerant to this drug. And another aspect that I want to bring to your attention is also this heteroresistance. And heteroresistance, as described in bacteria, is basically a phenomenon when the azogenic population exhibits a range of drug susceptibilities. And somewhat similarly, in Candida albicans, for instance, Gautier et al. Describe similar phenomenon where the cells, rather than showing some kind of uniform tolerance levels across the zone of inhibition, they show those outstanding colonies. As we can see here, some colonies are tiny, some colonies are bigger. So there's that heteroresistance that is happening, and the nature of those colonies, whether they are fully resistant or just can simply survive transiently those exposures to fluconazole, that is a secondary question, but those phenomena are also described in fungi. In Cryptococcus neoformans specifically, the term heteroresistance was defined as well, but it refers mostly to those cases where Cryptococcus neoformans can, in a stepwise manner, kind of become actually resistant to the drug, and typically forming resistance that is twice as much to the original exposure. Like usually we are talking about the MIC exposure, then those cells colonies pop up that are now twice as much resistant. And it's type of acquired aneuploidy, because the primary mechanism in Cryptococcus neoformans is really the formation of aneuploidy, and which is somewhat reminiscent or common to also Candida albicans, where the aneuploidy can form and indeed result in the increased levels of ERG11, which is the target of fluconazole, which is the key enzyme in ergosterol biosynthesis. So, of course, elevating the levels of ERG11 will make cells resistant to some degree to, if it's a roughly and one-yet copy, then that really increases the level of resistance twice. But also there are other genes that may get elevated in terms of their levels, like for instance efflux pumps. And I will switch back now to Candida for this one, for this example, just to illustrate how basically an example how treatment. Yes.
To conclude, sorry.
Yes. So I'm almost done. I just show the example where or maybe I will skip and go to the outstanding questions, because that's what I would like to just emphasize, is that they are still outstanding questions that exist in the subject, and I would like to just refer you and all the viewers to the fact that there are many outstanding questions and unanswered things, and hopefully we will have a chance to discuss it. And I apologize for going a little bit. I guess I'm too excited about the subject, and that's why I'm going a little bit outside of my time frame. But I just want to acknowledge the co-author of the study here, the Dr. Judith Berman from Tel Aviv University, and the lab members that I have currently in my lab, and the funding sources. Thank you very much, and I apologize for going a little bit over time.
Yes, thank you very much, and I'm sorry for rushing you. That's just a bit run over the time. So, so thank you again, Łukasz, for this engaging presentation, and we really appreciate your insights. I can see that some more questions have come up for your lecture, and you can, as I said, just try to answer to at least some of them in the chat window, but we may get back to some during the discussion at the end of our webinar. But now let's proceed to our next lecture and next speaker. So in fact, we have two last lectures, which both will be delivered by two bioinformaticians, who, I will be frank, are among the most outstanding, best I have ever had a chance to work with. And they both deal with tuberculosis. So as the first one, we will have Dr. Michael Auger from the Integrated Mycobacterial Pathogenomic Unit at the Institut Pasteur in Paris. And Michael, as I said, is a bioinformatician, and he's mainly focused on deciphering mycobacterial evolution, with a particular interest on Mycobacterium tuberculosis. And his talk will thus present some findings on key events from the past that seem to have shaped Mycobacterium tuberculosis to become the dominant pathogen, which is still nowadays. And so, Michael, I'm passing the floor to you, and it's a great pleasure to have you with us.
Thank you, Tomasz, for the introduction, and thank you to the FEMS organizer to give us the opportunity to present the work that we perform in the lab. So we are going to do a time travel by introducing some key events that occur through the evolution of Mycobacteria, leading to the emergence of Mycobacterium tuberculosis, the causative agent of tuberculosis, which is still a major human health threat still nowadays. So as a very broad view, for a start, let's look at the tree of life. So Mycobacteria belong to the Terrabacteria, and they belong to the phylum of Actinobacteria. And they are characterized by having their own family, which is named the Mycobacteriaceae. So overall, Mycobacteria are aerobic, generally non-motile bacteria with a rod shape that is depicted here with a picture of Mycobacterium tuberculosis. And they are also considered to be gram-positive. However, this is not as simple as this. So bacteria are classically divided into gram-negative and gram-positive, which is based on the color that they obtain after a gram staining. So the gram-negative, which are referred as diderm, are the cell envelope that is composed of an inner membrane, a thin layer of peptidoglycans, and an outer membrane. While for the gram-positive, that are referred as monoderm, they have an inner membrane, a thick layer of peptidoglycans without an outer membrane. So as I said, Mycobacteria are considered to be gram-positive. However, if we look at the composition of the cell envelope with the inner membrane, the layer of peptidoglycan, a layer of arabinogalactan, the outer membrane, which is composed of mycolic acids, and on top of that, a capsule, it actually looks like more to the structure and the architecture that is found in the diderm bacteria. However, the outer membrane, which is also named as the macromembrane, has an architecture that differs evolutionarily from the outer membrane of the diderm envelope, which raises the following question: did it arise from the remodeling of an existing outer membrane or de novo? And for this reason, Basil Bobenaya and co-workers actually proposed the term of neoderm envelope for the Mycobacteria to characterize this distinct structure and also to distinguish them from the diderm and the monoderm. So now we go into the evolution of Mycobacteria at the genus level. So there are about 250 species that has been defined according to the taxonomy and for which you have a phylogenetic reconstruction that is depicted here. So overall, Mycobacteria are usually classified according to two criteria. One of them is regarding the time that you need to wait to obtain colonies on agar plate. So we define the rapidly growing Mycobacteria, for which you will need a couple of days, up to 1 week, to obtain colonies on agar plate. For example, this is Mycobacterium smegmatis, which is a non-virulent Mycobacteria, or Mycobacterium abscessus, which is an emerging opportunistic pathogen. On the other hand, you have slow-growing Mycobacteria, for which you need to be more patient, as they will require a couple of weeks or several weeks to obtain colonies on agar plate. And two examples of them are the Mycobacterium tuberculosis and Mycobacterium caneti. Another way that we use to classify Mycobacteria is related to the disease. So we distinguish the tuberculosis causing Mycobacteria from the non-tuberculous Mycobacteria. It's actually quite simple for the tuberculosis causing Mycobacteria, as there are only two, M. caneti and tuberculosis, while all other Mycobacteria are non-tuberculous. So the definition of Mycobacterial species is based on classical information, the sequence of the ribosomal RNA, the spacer sequence, and housekeeping genes. And overall, we usually define Mycobacteria as a single genus with this Mycobacterium. However, some years ago, it was proposed to split the genus into 5 genera that are depicted here. Although this caused quite some debate in the community, as well as some confusion in terms of diagnosis, the choice of treatment, and it was also not very well supported by data. So more recently, it was proposed that the genus should be split into 2, Mycobacterioides with an M. abscessus, and mostly, and all the other clades defined as before as Mycobacterium. One example of interesting evolutionary events that occur at the genus level in Mycobacteria is related to the type 7 sequestration system, also referred as ES6. So the type 7 sequestration system represents a molecular nanomachinery that lies within the inner membrane, and that is composed of several proteins, which has been structurally defined. And they allow the export or the transport of ES6 and ES6-associated substrate through the membrane to the extracellular environment. So there have been 5 ES6 secretion systems that has been observed and defined, which are numbered from ES6-1 to ES6-5. And it has been considered that the ES6-4 secretion system, which is a smaller one actually, is the ancestral. And the reason for that is because it's the only one that is found across actinobacteria, such as Rhodococcus, Nocardia, and Corynebacterium. And it seemed to have been derived from the FTSK gene cluster that is found across Mycobacteria and also in Firmicutes. And the reason why it's considered as the ancestral, it's because all the other ES6 secretion systems, 1, 2, 3, and 4, 5, are the only one that I found within Mycobacteria. And it seems that they have been gained through evolution at different waves, following, for example, gene duplication, gene diversification, and horizontal gene transfer. And one of the key components of the integration of the different various ES6 secretion system is related to the ES6-1, which is one of the major virulence factors of tuberculosis causing Mycobacteria. So now if we look at the tuberculosis causing Mycobacteria, well, how did they emerge from non-tuberculous Mycobacteria is quite unknown. So far, the most closely related non-tuberculous Mycobacteria to the both tuberculosis Mycobacteria, M. tuberculosis and M. caneti, is related to M. decipiens, which represent a human opportunistic and slow-growing Mycobacteria. While it shares some features with tuberculosis causing Mycobacteria, there are also many differences in various aspects. And to fill the gap between how this tuberculosis causing Mycobacteria have emerged, this is quite unknown. And we can hope with the future on identification of new isolates and new Mycobacterial species that will lie in between, that will help to decipher how did they emerge. So I would like now to only focus on tuberculosis causing Mycobacteria. And as I said, they are separated and considered within 2 clades, Mycobacterium caneti which has been isolated in 1969 for the first time by George Canetti, and the complex of Mycobacterium tuberculosis, which has been isolated for the first time in 1882 by Robert Koch. So they cause the tuberculosis disease, but however, they are distinguished in many different aspects. One first aspect is related to the number of cases caused by this bacteria. So M. tuberculosis causes more than 10 million cases, new cases every year, resulting in more than 1 million deaths. And the lineage of M. tuberculosis are spread almost all over the world. By comparison, by contrast, M. caneti strain, there are less than 100 isolates that has been obtained so far, and they are restricted to TB patients that are mostly from or with connection to Djibouti and the Horn of Africa. Another important aspect is related to the virulence. So the blue line corresponds to Mycobacterium tuberculosis, while all the other lines correspond to various M. caneti strains. And then you can see here, in comparison to M. tuberculosis, the M. caneti strain depict a similar multiplicity during the acute phase of infection, but they depict a lower persistence in a later stage of infection. And if we look at the histopathological level, M. caneti strain depict less intense lung lesion and inflammation in mice in comparison to M. tuberculosis. Another main difference between these both clades is related to the colony morphology. M. tuberculosis represent a rough morphotype, while M. caneti strain has a smooth morphotype. And this variation of morphotype within Mycobacteria between smooth and rough has been associated with cell surface glycolipids, such as glycopeptidolipids for M. avium, M. abscessus, or lipolysaccharide loss for M. kansasi and M. marinum. So we could bring some insight on how the switch seemed to have occurred between the smooth to the rough morphotype, as we obtained for the strain K in the past of M. caneti, a spontaneous rough morphotype in the lab. And when we look at the genome sequence in comparison to the smooth morphotype, we actually could observe that there was a recombination event between the two copies of the PKS5 genes that were merged into a single copy, and that also resulted with the deletion of the PAP gene. And this recombination event was linked to the loss of LOS biosynthesis, which could not be observed anymore in the rough morphotype in comparison to the smooth morphotype. And if we complemented this rough morphotype with an intact PKS5 locus, then we could obtain again the biosynthesis of LOS. While in comparison for M. tuberculosis, whether you complemented them or not, they are still incapable of producing loss. And it was also a subsequent surface remodeling, as the rough morphotype could be reverted back to the smooth morphotype. And most importantly, this rough morphotype of M. caneti strain K depicted an increased survival and growth during infection of THP1 macrophages, which was similar to the level that is obtained for M. tuberculosis and much higher than the one that is obtained for the smooth morphotype. Another aspect, if we look at the genome sequence of M. caneti strain versus the M. tuberculosis complex strain. So overall, M. caneti and M. tuberculosis depict a very similar whole genome organization and high synteny, which is observed here with at the whole genome alignment between different strains and the M. tuberculosis complex strain, M. tuberculosis and M. bovis. However, the genome size is much higher, which can be up to 100 to 200 kb larger than the size range of the M. tuberculosis complex genomes. In addition, there is also a much greater genetic variability within the M. caneti clade in terms of polymorphism signals, such as SNPs, insertion, deletion, and as well number of genes. And this exemplifies here for the number of SNPs, as this range can go from 6,000 to 60,000 when comparing different caneti strain versus M. tuberculosis, while if you compare two members of the M. tuberculosis complex, you will obtain less than 2,500 SNP. And finally, which is actually linked to the greater genetic variability within the clade, they actually depict a recombinogenic population structure that actually is distinguished from the clonal expansion that occur within the M. tuberculosis complex. So chromosomal DNA transfer in Mycobacteria has been mostly described for M. smegmatis, It requires prolongated cell-cell contact. It is mediated by homologous recombination, and it is regulated by the TAP7 secretion system. And this type of transfer was defined as distributive conjugal transfer, and is result in mosaic genomes with transfer of multiple unlinked DNA segments with independent location. So we actually wonder whether this type of transfer actually could occur in Gdansk, and that could be linked to the recombinogenic population structure. So we developed an experimental model which consists of the donor strain and a recipient strain, each carrying a different antibiotic-resistant cassette. And if the transfer will occur from the donor to the recipient, then we will have recombinant that will be resistant to both antibiotics. So we have made in various mating assays by using as donor strain various members of the M. tuberculosis complex, and as well various M. caneti strain. And for when we're using the L strain as a recipient strain from M. caneti, for all of the examples that we try mating assay, we could obtain a recombinant, which suggests that tuberculosis-causing Mycobacteria can successively act as donor strain in chromosomal DNA transfer. And for the capacity of being recipient in chromosomal DNA transfer, well, it was not that simple, as most of the all the members of the complex that we have tested as recipients were unable to receive DNA, while within the caneti strain, it was a bit variable. Some were not able to receive DNA, while others, such as the G, the H, the I, and the L, were capable of receiving chromosomal DNA transfer. And when we sequence the recombinant, this is actually what we could observe, that we could observe the transfer from the DNA fragment from the donor strain to the recipient that was then found in the recombinant genome. In the term of mechanistic, we know that in smegmatis, chromosomal DNA transfer is mediated by both ES61 and ES64 type 7 secretion system. However, in M. caneti, which appears that the transfer is independent of the ES61 type 7 secretion system. So highlighting some differences that exist between non-tuberculous Mycobacteria and tuberculosis-causing Mycobacteria. And we are still currently investigating whether the ES64 type 7 secretion has a role in mediating chromosomal DNA transfer. So for all these reasons that I've explored and exposed, M. caneti actually represents a very interesting lead to study the emergence of M. tuberculosis complex, as it provides a link between both. But for the reason that the bottleneck that appear to go from a recombinogenic clade to clonal expansion is still unknown, but we can only assume that the M. tuberculosis complex emerged from M. caneti-like ancestors. And finally, we are going to go into the M. tuberculosis complex. This is schematic representation of it. It is composed of two main type of lineage. On one hand, you have the human adapted strain, which represents the lineage from L1 to L10. And then you have also the animal adapted strain, which are capable of infect various animal species, not only the one that has been depicted here, and in very rare cases to also cause infection in human. So all of this lineage has been associated with some particular region of the world, but basically, as you can see on this diagrams, that M. tuberculosis has spread all over the world, and most of the strains actually that are identified and observed and isolated represent the lineage 2, 3, and 4. Although this is not completely true, as the most dominant lineage is the lineage 1, which is mostly found in India and the Philippines. So given the geographical spread of the M. tuberculosis lineage, it was assumed that the M. tuberculosis complex has an African origin. As a reason for that is because M. caneti, the closest known ancestor of the MTBC, is mainly isolated in the Horn of Africa. There is also the presence of all human adapted lineage, but also the 6 lineage that are restricted to this continent. And there's also the presence of the mammalian species that are associated with infection of animal adapted strain. This for the reason that it was assumed that the emergence of the M. tuberculosis complex occurred 70,000 years ago in Africa. And then it was followed by the Neolithic expansion, growth of culture, animal domestication, adoption of agriculture 10,000 years ago. And then it was back to Africa again about less than 1,000 years ago by different waves of exploration, trade, and conquest that will explain why the different modern lineage are now also found in Africa. But more recently, there has been the isolation of ancient DNA samples from human remains that were dated between 250 and 1000 years ago, and which actually help to identify that the most recent common ancestor of the MTBC seem to have emerged less than 6,000 years ago. So one last point about the complex of M. tuberculosis regarding the different lineage is that there was some region of difference that were lost throughout the evolution. And for time reason, I will only present you one of the particular lineage, that is the TBD1, which distinguishes the modern lineage that we found nowadays all over the world in comparison to what we call the ancestral M. tuberculosis lineage. So this TBD1 region actually is located within the gene MMPS6 and MMPL6. The MMPS and MMPL proteins are involved in the transinner membrane transport of lipid residue, and the TBD1 region, the loss that was observed resulted in the loss of the MMPS6 genes and a shorter version of the MMPL6. And what we could observe actually is that this loss of this region increased the virulence of the strain that benefits from this loss in comparison to M. tuberculosis strain that has a TBD1 region that is intact. It was observed at the wild type level, but also by generating different mutants. For example, here, if we remove the TBD1 region, then the virulence was increased in comparison to the wild type. So the reason of this enhanced virulence was associated with an increased resistance to oxidative stress and hypoxia, which are two important mechanisms to control the infection by the host. And with this, there are very numerous people that need to be thanked from the lab of Roland Broche, but also numerous collaborators who contributed to the work I've presented you today. We have also our financial support, and I also provide you the link for further reading, as there are many other aspects that I did not have time to introduce you, which is some part that we wrote in our review, but also from other lab, the Sebastian Gagne's lab, for example, who also contribute to explore this evolution of M. tuberculosis complex. And with this, I thank you all for listening.
Thank you very much, Michael, for this beautifully delivered presentation, and it was really inspiring, as is evident also from some questions that have come in. So yes, so once again, thank you very much. Yes, so problems with my, okay. So now we will have our final speaker. So it is my pleasure to introduce Dr. David Cuvin, who is, as I said earlier, bioinformatician and researcher at the Institut Pasteur de la Guadeloupe, as well as associate professor at the Université des Antilles. And he specializes in bioinformatics, contributes to training and supervision of bioinformaticians, and his research focuses on bioinformatics applied to molecular epidemiology, with particular emphasis on genomic sequencing, metagenomics, and phylogenetic analysis of mostly pathogens, and particularly Mycobacterium tuberculosis. So please, David, it's your time.
Thank you, Tomasz. Thank you all organizers for this opportunity to present our work on molecular typing of Mycobacterium tuberculosis. I don't know if you see my slides.
Now it's perfect. Yes, it's a full screen mode. Yes, great.
Thank you very much for the invitation. So I will talk about molecular typing of Mycobacterium tuberculosis. So the work we done recently for the FEMS journal. So I will also, like Michael, I will talk about tuberculosis too. So as you know, Mycobacterium tuberculosis causing tuberculosis is still an important health issue worldwide. So it is important to develop tools and databases using molecular typing to better understand and better control the disease. As you know, my molecular typing and online database allow to better understand transmission chains, outbreaks, and the databases can also help us to store data like genotypes, genomes, or phenotypes data. And it also helps us to better compare strains across regions, across different regions. There are different techniques, different molecular techniques available worldwide. So some techniques are better used and better are easier to use than others. For example, the IS 6110 RFLP technique based on DNA fragments is highly discriminatory but very technically demanding. However, techniques like polygotyping are fast and easy to perform, but with a lower discriminatory power as compared to RFLP. And Miriam typing is also a technique widely used. It provides also a standardized format. Now, let me talk about databases. So, development of databases has begun since the 1999 years in Guadeloupe, a little French Caribbean island. And there are some different versions of the database. So the database is still ongoing today. It's still in development and it's still growing. So now we have, for example, the classical genotyping data of about 130,000 isolates or profiles. So here is our location of the lab in the Caribbean. And to talk about the recent SIDV2 version, we can see some distribution of spolygotype families worldwide. As you already know, even the molecular typing methods I talked about earlier are now being replaced by whole genome sequencing. Still use and we see energies to attack based. We can see distribute these and in so sequencing is now the gold standard. The gold standard for studying Mycobacterium tuberculosis, but also other pathogens. So now we are trying to basing on these whole genomes to do our analysis. For example, we extracted more than 7,000 genomes in the NCBI RefSeq database to compare with what we already have in classical genotyping and to make some comparison with data we already have also in the CIDVIT database. So now it rapid TB families and drug resistance maps in Europe, for example, and we can see some families, some patterns are being well represented, such as Euro-American lineages. Now we are trying to retrieve and simplify genomic information using whole genome sequencing. So we have now complete genomes, complete information, so we can do some analysis basing on the specific genes or specific sequences, and we are trying to add to use some AI tools to better understand data, to better compare also this data. We are also using bioinformatics tools like PyGenomeViz to search for similarities between some genomes. For example, here we have some drug resistance genes that are well known in Mycobacterium tuberculosis. So we can compare them in various genomes. Okay, and now with the advent of AI, we can use some machine learning to compute the data, the whole genomes from either Illumina, Nanopore, sequencer, or other. And we are trying to also do some we are using some techniques, some algorithms like decision trees or others to better understand this data. And now, for example, with decision tree, we have some better explainable AI to better understand regarding the genes, the presence, for example, or absence of genes, of specific genes, to determine the lineage, for example, of Mycobacterium tuberculosis complex. So some results with also random forest were very, very good, for example, to classify Euro-American lineages, but also EAI or other lineages. And we can also now use pre-trained encoder from transformers to better classify DNA sequences. We have also an approach with convolutional neural networks in development and some classification of sequences provided real nice scores of for the classification of the genomes of Mycobacterium tuberculosis. So thank you very much for your attention. I have some takeaways messages regarding molecular typing. I think that molecular typing is an essential for TB control. And as you know, whole genome sequencing is now the most powerful approach, and we still need some data integration and analysis to improve global health response. Thank you very much for your attention. And I would like to thank all collaborators of these studies, but also all collaborators in Guadeloupe. Thank you.
Okay, thank you very much, and thank all the speakers. And now we have a question and answer sessions. So I see many questions in the special window, and so of course we'll not be able to discuss all of them because of the time restriction. So I will choose some questions. If the speakers can answer some of them in the meantime, you are welcome to do this in this special box. But let's start and discuss. Okay, so first question: what are your experience using MinION versus Illumina for Mycobacterium tuberculosis?
Sorry, is
It was addressed to Mikkel or David, perhaps to Mikkel.
okay.
Sorry, I didn't know, Grzegorz.
Yeah, well, it is not indicated, so anyone who can answer this, who is using Illumina and MinION.
Well, so we do not have a lot of experience with nanopore sequencing for M. tuberculosis, has widely been more used for Illumina sequencing. But actually, they have some pretty good result with a nanopore, as a big issue with the sequencing of M. tuberculosis genome is related to the IgC content, which is quite complicated for short-read sequencing and the Illumina technology. While actually the nanopore technology better overcomes this issue. So we do nowadays a few sequencing using the nanopore, and it works pretty well. This, I think, becoming more and more interesting to use long-read sequencing with nanopore technology, especially that the quality is very good now. So there are some people that are actually considering to use it as well for variant calling. So this is pretty good advance for the sequencing overall of mycobacterial genomes.
Okay, thank you. And another question to Mikkel. Can you advise which cell can be a good model of bacterial mechanism study between Caneti and Smegmatis?
So by cell, I assume the person is talking about host cell. So I will answer this question considering this. Well, for Smegmatis, it's a non-virulent pathogen, non-virulent bacteria. So I'm not really sure that's very feasible to use some host, or at least I'm not very aware of. Now the question is related to M. caneti or M. tuberculosis. They have several cell culture model that has been used. Most of them are related to monocytes that are then derived into macrophages. So they are the immortalized cells with the THP1 from human macrophages. There are also some work that is done mainly from blood-derived monocyte, which has also converted into differentiated into macrophages, and that's quite interesting cell model because that brings the donor viability. But also people work on the mouse cell line that are, for example, the bone marrow derived monocyte. I don't remember the exact name of these cells. You have also alveolar macrophages. So they are, that's, I think, to my knowledge, the most used cell culture model for investigating such interaction between the host and M. tuberculosis or M. caneti.
Thank you. And now the question about the Pseudomonas. What are genetic mechanisms are likely responsible for the solvent resistance in Pseudomonas, especially in respect to the contribution of efflux pump in the resistance, and how does the effector molecule influence TTGR DNA binding activities?
Okay, thank you for the question. Well, the solvent tolerance is not a single element that is involved in the response. It's a number of elements. The one that gives this extremely solvent tolerance key is the TTGHI pump. And what happens is that this pump is plasmid encoded, and the plasmid is self-transmissible and can be going from one Pseudomonas to another Pseudomonas and provide solvent tolerance. The genes of the pump are always switched off in the absence of solvents. Once the bacteria see the solvent, a little bit penetrates into the cells, interact with the regulator, and the regulator is a repressor, so the complex regulator effector molecule release the repressor from the DNA, and the pump is present. That is the main mechanism behind solvent tolerance.
Thank you very much. So coming back to tuberculosis, another question about this. Can known tuberculosis-causing microbacteria become an opportunistic bacterium that causes tuberculosis?
Well, that's I'm not sure we understand the question. It seems indeed that there is an opportunistic mycobacteria that turn into tuberculosis-causing mycobacteria at some point regarding the evolution. But this is not have been achieved yet to identify what is this ancestor. And then regarding the tuberculosis-causing mycobacteria, they are virulent. They also might originate from an environmental reservoir, but same thing again. We have no clue about that, only assumption. As long as we do not identify the missing link between the non-tuberculous mycobacteria and tuberculosis-causing mycobacteria, then we cannot really address this question. It's pretty much what can I say about it. I don't know if David have more ideas to address this question.
If I may, Grzegorz, just one word.
Yeah. Yes, Tomasz.
May chime in. This is a quite interesting thing because, as you perhaps most of you know, there are a lot of mycobacteria that are opportunistic pathogens, and these are those mycobacteria other than tuberculosis. But what's interesting is that some of them can imitate TB as the causing agents. I'm thinking of Mycobacterium kansasi, for example, which causes a TB-like, very disease, although being opportunistic pathogen. So this is really very tempting to all us to decipher this mechanism and basis for responsible for transitioning the pathogen as the true pathogen, obligatory one, yes, and to keep it or keep it as an opportunist. So I think it's still a large field for explorations.
Yes, and kansasi is actually quite regarding evolutionary distant from M. tuberculosis and M. caneti. So there must have been an evolutionary transformation which is found in other bacteria. I think that Salmonella is one example as well from environmental or non-pathogen towards the obligate pathogen. But for the field of tuberculosis, this has not been achieved yet to be deciphered. So we can only hope with the future to better address this question by isolating more and more new Mycobacterial species. So closer to the tuberculosis-causing Mycobacteria.
And there is another question very related to that one. Is there any other species of Mycobacterium that is as pathogenic to humans as Mycobacterium tuberculosis?
Yes, I think that maybe, I don't know if it's true, but Mycobacterium avium or Mycobacterium abscessus could be also dangerous ones. But I know maybe there are also other Mycobacterium.
Yes, you have Mycobacterium leprae, ulcerans. So you have other bacteria that are pathogenic, not only opportunistic. So there is, within the genus, there is quite wide range of pathogenicity. So it's quite wide, and also depending also on the host.
Great. Okay. So again, staying with Mycobacterium and the question to Michael. In the Mycobacterium tuberculosis tree, it is quite interesting to see segregation based on geography of and animals. What could be the possible reason for such a specificity?
So that's as well a very interesting one. Overall, regarding the animal species, so the name of the animal adapted strain that has been given from either the main animal from which they have been isolated, or also the first original isolation. But it's very wide in terms of mammalian species that they can infect. It's really a wide range of mammalian species. If there's something quite interesting in the tree is that at some point there is a clade of animal adapted strain that appear to be separated from the human adapted strain, but on the other hand, there is still a clade which was the lineage 6, 9, and 10, which are actually then emerge, and then you have another clade of animal adapted strain that is present. So it seems that the ancestor of the Mycobacterium tuberculosis complex, or the hypothesis that is in the field, was maybe more generalistic as we thought, and it was capable of adapt to various hosts. And once it is adapted to this host, it was very adapted to it and without any change, because the switch from animals to human seemed to have appeared. But basically, the human adapted strain, they very rarely, if not, but I think it's rarely, can cause infection in animals. And for the animal adapted strain, there are some cases where they infect humans, but it's rather look like spillover, or like some people that are highly exposed to the bacteria from the animals. And also, it's in some cases where the patient are immunocompromised or incompetent. So that will actually explain how the pathogen managed to infect a new host. And then regarding the evolutionary history, so I mean, the oldest one of the oldest isolates that were obtained were like the Mycobacterium pinnipedi, which infect sea lions and I forgot the name of this organism, seals. There is some hypothesis that some people get contaminated, the population that actually eat the seals, and that's how the bacteria could switch from one host to another one. But it's it's highly discussed, and there are many hypotheses, but there are still many things to address to fill the gaps, definitely.
Okay, thank you. Now, very general question, perhaps to all speakers, because it's or to each speaker. Quite interesting. What emerging techniques in molecular microbiology do you think will have the most impact in the next 5-10 years? Well, hard to predict, but maybe you are trying to use something very new. Okay, who
I
would
think
like?
maybe I can relate to this. I think from my perspective, it seems like any techniques that acquire a lot of data, large sets of data analysis with the use of AI, probably it's going to be the big, huge game changer in the future, including things like single cell sequencing and things like that. So at all sorts of levels, and recognition of different patterns and gene expression patterns using the AI. I think this is definitely going to be growing field, and is already like quite impressive, as we all know.
Yeah, that's the question, because in eukaryotic genetics, the single cell techniques are becoming or extremely important. Do you think the same we can expect for microbial cells?
I think it's a little bit more difficult because of the amount
Yeah,
of
it is. This is why I'm asking.
But I agree with Luca that big data and artificial intelligence will make a difference. And also, all our presentations have been related, let's say, to single microorganism. And I think more and more we are going into microbiota or microbial communities because there is not a single cause for a disease. It's a combination of effects. So I think we will make big progress in understanding interaction between microbes in different niches. I think that will be really relevant. The approaches of molecular technique, combination of optimetrics, deep sequencing, and all of that will be running very fast.
Yeah, indeed. So it appears that there are two directions. One in the single cell, so very deeply going to the molecular levels, and then to investigate the communities of bacteria, like microbiomes and so the global picture. And so I think that there are two quite different, but actually very important ways to investigate microorganisms to get the bigger picture. Okay, well, the last question, and again, very general, but can you compare pathogenicity of Mycobacterium tuberculosis versus Pseudomonas aeruginosa? That's again the more general question, because the comparing two very different bacteria is not easy, because the mechanisms are different and the lifestyles are very different.
Well, I'm not an expert in Mycobacterium, but what we can say about Pseudomonas is that they are not really virulent microorganisms. Or Aeruginosa is different, of course, but I'm talking more broadly because I'm not an expert in Aeruginosa. But what we can say about Pseudomonas is that they are not really virulent microorganisms, but they can resist to. They have antibiotic resistant determinants because it's like a co-resistance. They use other mechanisms as well, as antibiotic resistant determinants. Although some strains has some plasmids that accumulate a lot of these antibiotic resistant mechanisms, but I think they are quite different. Also, Pseudomonas are environmental strains. So I don't know. I think they are quite different, but maybe the other panelists can talk more about Mycobacterium.
Yeah, well, as we heard a lot about them, but it's they are very different. So that's that's very hard to compare with which is more or less pathogenic because the mechanisms are so different. Okay, well, the time is going, and I think we can reach the last stage of our webinar. So discussion is closed now, and I would like to ask Tomasz for the concluding remarks.
Sorry, I just thought about, and there was one question some time ago, very similar to one I wanted to ask Łukasz. We do have just
Okay.
1 minute.
So the last question.
Also a bit general, but as you all know, all attendees and speakers, of course, there's a very hot discussion nowadays as for the nomenclature of fungi. So in the context of your presentation, Łukasz, showing the growing, in fact, heterogeneity in terms of genome and phenotypic composition, how do you think this influence this ongoing discussion? Do you see any chance to produce a consensus or in terms of naming the fungi, because there is let's frankly call it mass in this fungal nomenclature. So how do you see it, and do you think that we should rather be guided, we are scientists, by the genetic findings or phenotypic findings, or how, in some way, just to merge them and as I said, to work out any compromise or consensus? What do you make of it?
Well, definitely genetic. Obviously, the nomenclature that has, a very long history of systematics of all sorts of organisms dates back to times where genetics was not really clear, and as we can expect that more and more information that comes from genomes and insights into surprising relatedness of things that morphologically are very distinct and very deceiving, let us discover that things are actually the same thing or related. So I think definitely genetics has to be the subject or the factor that will really determine the actual relatedness of species and will influence the nomenclature. And I agree with you that there is a lot of inconsistencies and with fungal species, like for instance Candida glabrata, one prominent example that was renamed recently, and this is just like reflecting that it's going to be an ongoing process, gradual process, because there's some kind of resistance, obviously, and lag that is unavoidable in a way because of the vast literature on those subjects, including that, let's say, incorrect nomenclature. It's not incorrect, but it's like not strictly accurate. And then I think it will be a gradual process of improving it. I think my colleagues can agree that the same probably is true for bacterial species.
It's in medical mycology. It's very complicated. It's particularly complicated, I would say, and very many clinicians are so used to these traditional historical names, and when we further exploring the genomes, fungal genomes. The genera currently known are split again and again. So we have new species and subspecies, and then so it's really a big, huge problem, I would say, for clinicians, for not perhaps researchers, but for clinical practice. So this is that I think we should really keep in mind and discuss and to talk it over and over.
Okay, thank you.
Thank you.
Okay, so I have to close the discussion because of the time constraints, and I'm sorry to the people who ask questions which cannot be answered. We still have some time to answer by writing. So now, please, Tomasz, provide some summary and conclusions.
Exactly. So just again, we all know that this short discussion will not exhaust our questions and doubts. So perhaps I encourage all attendees to perhaps directly reach out to our speakers by email simply. So to conclude, I would like to warmly thank the speakers, of course, for their excellent, brilliant presentations and discussions and for their time dedicated. Of course, the attendees for coming to this webinar. I'm really happy that we could gather over 400 in one moment, perhaps. Grzegorz, did you see it? Or 300 from over for sure. It's close to or
several hundred, let's say.
So it's really great turnout. So thank you very much. Thank you for your questions. And also in closing, I would like to thank the organizers of this webinar and especially Erwin Rippmeister, FEMS managing editor, who was truly at the heart of this event and made it happen. And also I would like to give my special thanks to Joseph Shuttleworth, who was and still is with us on backstage for his excellent technical guidance and for managing all this technical side of this webinar. And finally, well, yes, I just once again I thank everyone and hope to see you next time or another occasion. And I hope I myself did not disappoint you. It was in fact my debut in this role as chair of this of such an event. So thank you and sorry and apologies for any technical issues. We had some during David's talk, but I think it was overall a very nice endeavor and adventure. So and will benefit all from today's meeting. So with this, again, thank you very much. Thank you, Grzegorz, for
thank you
sharing and lectures and attendees once again. Thank you very much.